Today I will talk to you about AlphaFold 3 was developed by Google DeepMind and Isomorphic Labs. It can predict the complex structures formed by proteins, nucleic acids, small molecules, and common metal ions, and can include post-translational modifications of proteins, including glycosylation, and chemical modifications of nucleic acids. AlphaFold 3 is the latest algorithm that can predict the structures of various molecules. AlphaFold 1/2 can only predict protein structures. There is also a corresponding database. AlphaFold 3 outperforms the best previous methods focused on a single task by a wide margin. It more accurately predicts the structures of proteins and protein-protein complexes, especially antigen-antibody complexes. Next, we will introduce how to use AlphaFold. The practical video is at the end. This is the molecule type. You can choose protein, DNA, RNA, ligand, ion. This is where the sequence is entered. Finally, click Submit. The best feature of AlphaFold Server is fast calculation and simple operation. If AlphaFold Server is used to predict a single molecule, then you only need to pay attention to the confidence. If AlphaFold Server is used to predict multiple molecules, then you need to pay attention to the score. In general, PTM scores above 0.5 indicate that the predicted overall fold of the complex is likely similar to the true structure. IPTM measures the accuracy of the prediction of the relative positions of subunits within the complex. Values above 0.8 represent high-quality predictions with confidence, while values below 0.6 indicate that the prediction is likely to fail. Hello everyone. Open the browser, enter AlphaFold Server, and click to enter the web page. There are generally two situations on the web page. One is https://golgi.sandbox.google.com, and the other is https://golgi.sandbox.google.com/about. 2. After entering, we can have an overview, and then we can look at the upper right corner of the page, which has the number of daily uses, and there are currently 20 times. Scroll down to the sequence input area and history records. You can see in the history records that the human DLD target and human ADAR1 target that have been tested before. 3. In this experiment, we first test the human DLD target, open a new search box, enter UniProt, enter the UniProt official website, and enter DLD to search. During the search interval, let's check the input area of AlphaFold 3. We can see that there are different options on the left side of the input options, such as protein, DNA, RNA, ligand, ion. The middle box can select the number of times the currently entered sequence appears. Ion and ligand support up to 50 copies, DNA/RNA/protein supports up to 1000 copies, ligand supports common ligands such as ADP, and ion supports common ions Mg2+, Mn2+. Common inputs have been introduced. And then we go back to look at the UniProt input results, select the human species on the left, and we find that there are still multiple results. We need to check carefully and select the correct protein. The UniProt ID selected this time, P09622, it takes some time to enter. We return to AlphaFold 3 to introduce the history record to everyone, where you can see the previous project name, submission or completion time, and the last menu option. If there was any input before, we can click Clear to directly clear the previously entered sequence. We return to the UniProt website corresponding to the previous DLD. We focus on 3 parts, one is structure, one is family and domains, and one is sequence and isoforms. We first click on structure and find that it is quite stuck, normal. Then we look at the sequence and isoforms option. We find that DLD has 3 isoforms. Be careful to determine which one you need. In this experiment, we choose the default first one, 509AA. You can also look at the remaining 2. We found that the 3 are only partially different in sequence. There is also an explanation above. This entry describes 3 isoforms produced by alternative splicing. We can choose the download option to download, or we can directly click Copy Sequence. After copying, the option will become copied. We can enter the corresponding sequence into the input box of AlphaFold 3. Then, we can look at the download interface we just clicked, and we can see the sequence display interface. After copying, we need to delete the return. Let's take a look at the structure of DLD on UniProt again. We can see that the protein as a whole is still relatively large. We can rotate different angles to view it. At the same time, we also found that DLD proteins mostly exist in the form of polymers. This time, we choose 5J5Z as the control template, select the RCSB PDB website, and enter the corresponding website. 4. Enter the PDB website. We can see the crystal structure of 5J5Z, which is a dimer. We can look down and see the corresponding protein information and small molecule information. There is an endogenous small molecule FAD in it. It should be noted that there is an amino acid mutation in this crystal structure, D444V, aspartic acid at position 444 mutates to valine. We can adjust the corresponding amino acids in the sequence just entered, find the corresponding amino acids, and then modify them. We will not adjust them in this practical operation. Use the default, copy set to 2, dimer, and then click Continue and Preview Job. 5. Here you can see the overall situation of the project. Let's modify the job name. The default is the time at that time. It is recommended that you name it according to the target protein. Here, name it according to name plus species plus PDBID plus remarks. After confirming that it is correct, click Confirm and Submit Job. After clicking Finish, you can find it in the history record and find that the project is in progress. 6. We select the previously completed DLD modeling results to view, and click Record to enter the result display interface. After entering, we can see the overall dimer situation, and we can view it from the perspective of publicity. At the same time, we can see that the protein as a whole has different colors, and the meanings of different colors are displayed on the structure, representing different confidence levels, such as dark blue represents confidence greater than 90, light blue represents confidence in the 70-90 interval, yellow represents confidence in the 50 to 70 interval, and orange represents confidence below 50. We see that the protein as a whole is dark blue, proving that the overall confidence of the structure is good. The figure on the right is expected position error, angstroms, which can be simply understood as the prediction error. The greener the color, the smaller the error. In this experiment, PTM equals 0.92, and a PTM score higher than 0.5 means that the overall prediction of the complex is similar to the true structure. The full name of PTM is the Predicted Template Modeling, and the full name of IPTM is the Interface Predicted Template Modeling, IPTM. In this experiment, IPTM equals 0.91. IPTM measures the accuracy of the predicted relative positions of subunits within the complex. Values above 0.8 indicate confident, high-quality predictions, while values below 0.6 indicate that the prediction may fail. The IPTM value between 0.6 and 0.8 is a gray area, and the prediction may be correct or incorrect. Judging from the IPTM score, the relative positions of the subunits in this experiment are relatively prepared. Both PTM and IPTM scores are derived from TM, the template modeling, scores. TM scores are very strict for small structures or short chains, so when there are less than 20 subunits involved and the PTM value is less than 0.05, PAE or PLDDT may better reflect the quality of the prediction. In general, the predicted structure is similar to the actual situation and has high accuracy. 7. Then we can select Download in the upper left corner to download the corresponding results. This experiment has already been downloaded, so it will not be downloaded this time. 8. Open the results we just downloaded. There are .json format files and .cif files. By default, the _model_0.cif structure is selected for analysis. If model_0 is not what we need, we can select other protein structure files for analysis. 9. The corresponding scores can be viewed in summary_confidences_0.json. We can click to see fraction underscore disordered, 0.04, has underscore clash, 0.0, IPTM, 0.92, number underscore cycles, 10.0, PTM, 0.92, ranking underscore score, 0.94. AlphaFold is sorted according to ranking underscore score. The larger the score, the higher the priority of AlphaFold recommendation. Ranking underscore score equals 0.8 times IPTM plus 0.2 times PTM plus 0.5 times disorder minus 100 times has underscore clash. 10. We compare the crystal structure 5J5Z of DLD with the model underscore 0.CIF just predicted. We open the PyMol software and drag the two proteins in, and we can see, the protein has been dragged in advance in this video. We display the crystal structure as a white cartoon and model underscore 0 in green. Select the align function in the action and compare